Hello everyone! In this video I'm going to be covering DNA structure, Mendel's experiment, and gene crosses. In the previous video I just discussed the intro to genetics, but I'm going to build off of that a little bit and we're going to go a little more in depth in these particular subjects. So let's get started. So in the last video we talked about cell division and how the chromosomes are passed to the baby through mitosis. But in this video, we are going to go more into the actual genes located on these chromosomes. So when you think of genes, what do you think of? Well, you might hear people say, you have your dad's eyes, or you have your mom's genes. These genes that show your eye color or hair color or diseases inherited are all coded by genes on the chromosomes. Now, you may remember in the last video how I said that when I say coded by genes, I'm referring to the sequence of base pairs in the DNA. So let's take a look at the actual DNA structure to understand the genes that are coded on it and inherited from your parents. DNA structure. So think of a ladder. Now imagine twisting this ladder to the right. This coiled up figure is the structure of the DNA. The two sides of the ladder, or DNA, are made up of sugar groups and phosphate groups, while the rungs of the ladder, or the parts that you walk on the ladder, are made up of nitrogenous bases. When there is a structure that is held together like this one, the parts are connected by tiny little bonds that are created by the molecular composition, or the molecular structure, of the parts. Here, the sugar and phosphate groups are held together by phosphodiester bonds. So they create the two long strands along the DNA. These alternating sugar-phosphate sequence is called the backbone of the DNA. Now for the nitrogenous bases, or the rungs of the ladder. These are between the two strands. These are parts of the DNA that actually do the coding for your genes. There are 4 kinds of nitrogenous bases: adenine, thymine, cytosine, and guanine. There are specific ways in which these bases like nitrogenous bases, can pair, and the order of these pairings in the DNA determines what the gene will be for a human. So some of the restrictions on pairing are that adenine can only pair with thymine, and cytosine can only pair with guanine. So adenine and guanine can't pair together, or adenine and cytosine can't pair, and etc. Remember how I was talking about how there must be bonds present to hold the structure together? Well, the bonds that are present between the base pairs, such as the adenine and thymine and cytosine and guanine, are hydrogen bonds. There are 2 hydrogen bonds between the adenine and thymine, and there are 3 hydrogen bonds between the cytosine and guanine. Okay, now that we know the structure of the DNA, we can see how the genes are actually shown on the chromosome. Genes on chromosomes. So remember last video we talked about chromosomes. Well, the chromosome of a child has 1 chromatid from the mom and 1 from the dad, and each of these chromatids has genetic information from each parent. So that means that you have 2 genes for 1 trait. Now you might be wondering, if my mother has brown hair and my father has blonde hair, do I get a mix of brown and blonde? Or, both my parents have brown hair, but I have blonde hair. How does this happen? This is because of the kind of gene your parent has, if it is recessive or dominant. So basically, there is a letter representation that shows the genes you have gotten, and these are based on the genes of each chromatid of the chromosome. Dominant genes are represented by a capital letter, and recessive genes are represented by lowercase letters. Also, a gene is said to be recessive if it is masked by the dominant gene. In other words, the part of the DNA on each chromatid that accounts for a specific gene is represented by any letter to signify if the gene is dominant or recessive. For instance, if you have a segment of genes that codes for blonde hair on one of your chromatids, and on the other chromatid, the same segment codes for brown hair, because you got brown hair from your mom and blonde hair from your dad, your genotype, or the letters that represent your gene color for hair, would be big B, little b. Again, capital letters represent dominant genes, so the big B would be brown hair, and lowercase letters represent recessive genes. So in this case, the small b would be blonde hair. So, because dominant gene is brown hair, it masks the recessive gene of blonde hair, and you would thus have brown hair. If you have blonde hair, but your parents have brown hair, on the other hand, this means that your parents are carriers of the recessive genes, meaning that they both have a blonde allele they can give you. And the only possibility for this is if your parents both have big B little b as their genotype and can give you the recessive little b. So your genotype is little b little b. So basically if your parents have big B little b, if your mom has that and then your dad also has big B little b, then they can each give you the little b and you'll have blonde hair because that's recessive and nothing is masking it. There's no dominant gene to mask the recessive gene. And so the recessive gene of the blonde will show in your hair color. But how exactly was it decided that blonde hair is recessive to brown hair? Well, Gregor Mendel, said to be the father of genetics, conducted a series of experiments on plants to decide this. Mendels Experiments. Mendels Experiments. He found that if he bred a true breeding tall plant with the genotype big T, with a true breeding short plant with the genotype little t, then all the plants in the next generation, called the F1 generation, would be tall. True breed, which is also called homozygous, just means that the plants receive the same kind, recessive or dominant, from both parents, so big T, and little t. So, if they received different genes from each parent, such as big T, little t, then this genotype is called heterozygous. So homozygous, heterozygous. Purebred, heterozygous. This can be so the experiment that he conducted with the big T crossed with the little t can be modeled through the use of something called Punnett squares. The Punnett square allows you to put one genotype from the mother on the top and the other genotype of the father for the same gene on the side, or vice versa. So using this tool, you bring down each genotype into the box and create 4 possibilities that the offspring of the mother and father could be. Just in addition, the 2 kinds of genes you get from each parent are called alleles. Anyway, in his experiment, with crossing the tall purebred or true-bred pea plant with a short true-bred pea plant, he found that the next generation, the F2 generation, has 3/4 of the plants tall and 1/4 short. Then finally, he found 3 things. One is that the 1/4 of the pea plants that were short in the F2 generation would produce only true short-bred pea plants in the following generation if they were mated with other short plants. 2, that 1/3 of the tall plants in the F1 generation would produce only tall plants in the F3 generation if they mate with the same genotype's plants. And 3, 2/3 of the tall plants in the F1 generation would produce 1/4 short plants and 3/4 tall plants in the F3 generation if they mate with the plants with the same genotype or self-pollinate. So using this experiment, Mendel basically discovered that this small purebred small t got masked by the genotype with a plant's genotype if it had a big T in it. And he found that by cross-pollinating all these different pea plants with different generations. Also, using the experiment, Mendel created the law of segregation. This just means that the 2 alleles that make up the gene from your mom, such as small b for instance, are broken down into big B and little b. And the same happens with the fathers, and then these determine the baby's genes. So basically, the law of segregation is that the genotype of your mom is broken down into the 2 alleles, the genotype from your dad is broken down into the 2 alleles, and then it determines the baby's genotype. And this can be shown through the Punnett square. The Punnett square we used here is for monohybrids. This is when you cross one gene and want to find the probability of the offspring to get that gene. Also, for these monohybrid crosses, there are two ratios you can use to display the information in the cross. One is the phenotypic ratio. This is the number of dominant to the number of recessive. The phenotypic ratio refers to what the plant looks like. Think phenotype such as physical. This is the physical appearance of the plant. So the phenotypic ratio is the number that appear dominant. It doesn't have to be purebred dominant, but just the ones that appear dominant, so that has a dominant or uppercase letter in the genotype, to the number of recessive, or the number that don't have any dominant genotypes to mask, dominant alleles to mask the recessive alleles. In the case of crossing the two parents of the parent generation with the pea plants, so the big T with the little t, the ratio would be 4:0, because when crossing, all the offspring have a dominant allele that masks the recessive, so there would be 0 for the recessive number, since the short trait is masked for all of the offspring possibilities. There's also the genotypic ratio for a monohybrid Punnett square. This is the sequence of numbers. It goes purebred dominant to heterozygous to purebred recessive. So for the same example, the ratio would be 0 to 4:0. Since there's no big T's possibilities for the offspring, and there's no recessive allele possibilities for the offspring, there's only heterozygous, which means one big T, one little t. In addition to monohybrid crosses, which display the crossing of one gene, such as here, whether it's tall or short, so the height, there are also dihybrid crosses and trihybrid crosses, which deal with 2 and 3 genes being crossed and the probability of the offspring getting 3 specific gene types. Dihybrid cross. Mendels Dihybrid cross. Here is an example of a dihybrid cross. This, as shown by the example, shows the probability for someone to get the gene that is represented by Y, and to get another gene, which is represented by R. Now this cross is asking to cross a big Y, big R, with a small y, little r. Unlike monohybrid crosses, where you are able to put the alleles for each parent on one side of the Punnett square, in dihybrid crosses, there's a little more work required. First, you need to find all the possible gametes each parent can give. You do this by essentially finding all the ways you can pair a gene for one gene and allele for another for each parent. So, for instance, in the all capital YYRR, you distribute the first Y with the first R, then the first Y with the second R, then the second Y with the first R, and finally the second Y with the second R. In this case, all the gametes produced by the first parent will be big Y, big R. And if you do the same distribution for the second person in the same distribution order, then all the gametes for this parent will also be little y, little r. But bear in mind that this will not always be the case for parents, such as for the gene big Y, big R, little r, will result in gametes of big Y, big R, big Y, little y, big R, and little y, little r. Anyway, these paired gametes for each parent are placed in the two sides of the square, and then the pairs are brought down to create a genotype for the two genes. This is similar to the monohybrid cross. This square, for instance, shows that the probability of the offspring to be yellow and round is 9/16, with the Y and the R representing the shape and the color. The representation of the genes as a photo and as observable, again, is not called the genotype, but the phenotype. Again, think of the phenotype with the pH like a physical appearance, with the pH in physical. So now there's a lot of information being shown in this Punnett square with the dihybrid cross, such as the phenotype and genotypes for the probability of the offspring. And this is simplified through the phenotypic and genotypic ratios. Dihybrid crosses, however, only have phenotypic ratios, whereas the monohybrid crosses you could pull lots of information such as the phenotypic and genotypic ratios. But this only has the phenotypic ratios. So the first number of the phenotypic ratio is dominant allele for both traits. Then the second number in the ratio is dominant for the first trait and recessive for the second trait. Then the third number of the ratio is recessive for the first trait and then dominant for the second trait. And the final is recessive for both traits. So in this instance, the ratio is 9 to 3 to 3 to 1. One way to make sure that you have the right phenotypic ratio for dihybrid crosses is if you add up the numbers 9, 3, 3, 1, it should add up to 16, which is the total number of possibilities for the offspring to have, or for the offspring to look like. So the offspring of the big Y, big R parent with the other parent being small y, little r, there are 16 of these options that it could be. So that was it for this video. I really hope you enjoyed and learned a lot about crosses, chromosomes, DNA structure, and how this all comes together. And I understand that there's a lot of vocabulary, but just while you're watching these videos, just note down the vocabulary and go through it because it's very important and will come up many times throughout the following videos. Please subscribe to this channel if you like these kinds of genetics videos, and check out some other videos I have on this channel, such as computer videos. I have a whole series on those, so check those out, and some biology videos and book reviews. If you have any comments for future videos, then leave those in the comments below, or if you have any questions for this video, then you can also leave those in the comments below. Anyway, here are just a couple of citations I used for the pictures of this video. Thanks for watching!